Hello, Lakers. We're going to start again. This time we're going to take a look at what factors affect enzyme activity. So I will power up my PowerPoint presentation here, and we'll get started. So the factors affecting the enzyme activity. Scientists like to look at what factors affect the rate of enzyme activity. In other words, sometimes enzyme catalyzed reactions can be speeded up or slowed down. How does that happen? What factors affect the rate of an enzyme catalyzed reaction? Okay? And so I came up with 5 or 6 things that definitely affect it. Let's take a look at them. We'll start with the initial substrate concentration. Now, the question is, what would happen if you increased the substrate concentration? So let's say I have a little graph here with the reaction rate, or the rate of the reaction that we're measuring, and this would be the concentration of the substrate if we get more and more and more of it. Seems like if you had more substrate, what would happen to the rate of the reaction? And you have a given level of enzyme. Well, it seems like it should go up. But does it go up forever? I want you to think about, maybe some of you have dogs. I have a dog. He can't catch popcorn in his mouth at all. He's terrible. He must have bad vision or something. But many of you have dogs that can catch popcorn. So if you have a pile of popcorn in your hands and you toss your dog a popcorn, your dog catches it. Think of your dog as being that enzyme. And the dog catches it and eats it. That's the reaction that happens. Toss your dog another piece of popcorn, catches it, and enzyme reaction happens. Toss the dog another piece of popcorn. Now, what if you increased the number of popcorn that you threw at your dog? So at first you throw just one piece, but now you're starting to throw one piece and then add another one. And then you throw one piece, quickly add another one, and a third one, and a fourth one. Well, the dog is pretty good at catching them and swallowing them. Chomp, 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 right? But at some point, what happens? You can be throwing your popcorn so fast that the enzyme, the dog, can't catch them all. There's a maximum rate at which your dog can catch popcorn. And so no matter if increasing the number of popcorns thrown isn't going to increase your dog's rate of catching them. That's sort of like enzyme activity. We don't have this like straight line reaction rate with increasing substrate concentration. We have something that looks more like this. So if we're looking at the reaction rate as a function of substrate concentration, at first we increase the substrate, we get an increase in the reaction rate, but we plateau. We reach a plateau. Why? What's happening? Well, take a look. At low rates, at low substrate concentration, we have low rates of the reaction because the enzyme has to find the substrate. It has to bump into it, right? As we increase the substrate concentration, we have a higher rate of the reaction because the substrate is bumping into the enzyme more quickly and the reaction's happening more quickly. You're feeding your dog more popcorn, right? But finally, at some point, the more substrate we add, we don't get increasing reaction rate. And that's because the enzymes that are present are doing their job as quickly as they can and are bumping into a substrate, reacting, releasing the product. Bumping into a substrate, reacting, releasing the product. Bumping into a substrate, catalyzing the reaction, releasing the product. Bumping, and they're moving as fast as they can. It doesn't matter how much substrate you throw at them. It doesn't matter how many popcorn pieces you throw at your dog. The dog can only eat so fast, right? So this reaction rate plateaus with increasing substrate concentration. We've reached the maximum velocity at which the reaction can happen, given the enzyme that we have. Could you do anything to speed up the reaction at this point? Yeah, sure. Sure you could. You could speed up the reaction by increasing the amount of enzyme you had. Absolutely, add more enzyme. And if you add more enzyme, we should be able to, the reaction rate should go faster. Yep, exactly. Okay, so we can look at how the initial substrate concentration affects the rate of the reaction. We get this plateau. And now, what about the speed of the enzyme, or the concentration of the enzyme? Absolutely. The speed of the enzyme. Enzymes have an inherent speed. Some enzymes have a higher maximum velocity than others. So if we're looking at the same enzyme in different species, maybe we're looking at this enzyme called peroxidase in turnips and bananas and apples. The Vmax of each enzyme, the maximum velocity, might be slightly different. And people who study enzymes love this stuff. Here's a third one, the efficiency of the enzyme. What does that mean? Okay, now suppose we have two enzymes. Maybe they're the same enzyme from different species. And we're looking at the rate of the reaction as a function of substrate concentration. And we see two curves for our two enzymes. One curve looks sort of like this. This would be maybe enzyme A. And another enzyme, maybe I'll do it in a different color here, color blue, has a different curve. It kind of looks more like this, but it eventually reaches about the same Vmax, if you will. That's our Vmax. Which of these enzymes is more efficient? Can you tell? Well, they have the same Vmax, but we might say that enzyme A is more efficient because at low substrate concentrations, which enzyme has the faster rate? Enzyme A does, right? At this low substrate concentration, the rate of the reaction under enzyme A is up here. The rate of the reaction under enzyme B is down here. Enzyme A is more efficient at grabbing the substrates and catalyzing the reaction. So sometimes we talk about the efficiency of the enzyme, if you will. And some enzymes are simply more efficient than others. So people who study enzyme activity are really interested in these factors. Okay, another thing that affects the enzyme activity would be cofactors, or non-protein helpers. It turns out that some reactions require cofactors. Cofactors are quite variable, but cofactors can include organic compounds like vitamins. A lot of our vitamins that we eat are cofactors, or inorganic elements like zinc or copper. And these cofactors need to bind to the enzyme so that the enzyme can function normally. If they're not there, the enzyme doesn't work. Here's a picture of an enzyme called polyphenol oxidase. This enzyme so the picture here is this three-dimensional structure of the protein. It's complicated, right? Now, this enzyme happens to be involved in converting catechol and oxygen into quinone, whatever. Actually, this enzyme is involved in causing your fruit to brown after you take a bite of an apple. You know how the browning happens in that apple, or you cut a potato and let it sit out for a while and it turns brown? This enzyme is responsible for that. It catalyzes the reaction that causes the browning. In lab, in the past at least, we've studied we often studied this enzyme and how it works. Now, this enzyme absolutely requires a cofactor of copper. Copper ions are required. They have to bind to this enzyme so that this enzyme can do its job. If there's not copper around, this enzyme can't work. It's a cofactor for the enzyme to function. All right, what other things do we have on our list? Oh, we have environmental factors. Environmental factors can affect the enzyme's activity. So when we think of an enzyme, remember that it has this active site or this catalytic center. Can you think of any environmental factors that might change the shape of the enzyme? And if you change the shape of an enzyme, if you mess with that active site at all, you're going to change the function. It's not going to function as well if you change the active site's shape. So are there any environmental factors that might change the shape of the enzyme? Yes, of course there are, right? Yes, there are. Think about temperature. Temperature can cause a protein to change shape. If you heat up a protein, the three-dimensional shape of that protein can fall apart. It can denature. And when the three-dimensional shape of a protein falls apart, so you've got this complex structure, and it falls apart into just like a long string of amino acids, its function doesn't work. It doesn't work anymore. Enzymes are proteins. So if you change the shape of an enzyme, it's probably not going to work anymore. Temperature can change the shape of the enzyme. And so what we've got here is the rate of the reaction, so the reaction rate as a function of temperature for two different enzymes. One of these enzymes comes from humans, and one comes from a thermophilic bacterium. What we see with the human enzyme, maybe it's the enzyme in your saliva called amylase. Who knows? It functions best at a temperature that is pretty close to your body temperature, right? So we get this, highest functioning pretty close to physiological conditions. And if we move away from that, if we get colder or we get hotter, we see a change in reaction rate. We see a dropping of it. So we see this nice curve. Right? The curve actually drops very quickly at a certain high temperature. That's when the enzyme denatures. The enzyme falls apart because of high temperatures, and then it can't catalyze the reaction. What's interesting about this graph is a second curve is on the graph showing an enzyme from a thermophilic bacterium. Lots and lots of bacteria live in the hot springs in Yellowstone National Park. So here's a picture of a couple of my kids a long time ago at Yellowstone National Park, and you can see beautiful colored pools. And these pools might be very hot, 75, 80, 90 degrees Celsius. You get to 100 and you're boiling, right? These pools are filled with bacteria and archaea and algae that are tolerant to heat. When we look at their enzymes function at much higher temperatures than do ours. So the optimal temperature for a thermophilic bacteria might be 77 degrees Celsius or 85 degrees Celsius. Their enzymes can stay stable and in a stable shape under hot conditions. But eventually, those enzymes can also denature. Okay, so that drop in the curve there is from denaturation. pH can also affect the rate of the reaction, or the rate of an enzyme-catalyzed reaction, because changing pHs can change the shape of the enzyme. If you change the shape of the active site, the enzyme can't function. So if we look at the optimal pH for pepsin, which is an enzyme that breaks down proteins in our stomach, and the optimal pH for trypsin, that's an enzyme that breaks down proteins in our intestine, we see they're quite different. Remember that stomach is very acidic. We have a pH of about 2 in our stomach, and sure enough, pepsin works best at about a pH of 2. If we move away from that pH of 2, we put pepsin in a pH of 4 or 5, it's not going to work at all. Its shape has changed. Its active site no longer can catalyze the reaction. But trypsin does much better at a pH closer to 8, 7, 8, 9. But when you move away from that, you see that the reaction rate drops off as well because that active site changes. Okay. One more situation. Inhibitors. Inhibitors can affect enzyme activity. What do you think they do? They inhibit the rate of the reaction. So they are going to slow reactions, right? They inhibit them. They slow them down. There are different kinds of inhibitors. So here we've got a picture of a purple enzyme. That's the protein, right? And the substrate it's catalyzing, and here's its nice active site. So the substrate binds to it, catalyzes it, great. Competitive inhibition is a type of inhibition in which you've got a molecule that competes with the substrate for the active site. So here, our competitive inhibitor binds to the active site of this enzyme. And when it's bound to the active site of the enzyme, can the enzyme function? No, the enzyme can't function, right? Because there's no active site available. Sometimes competitive inhibitors will bind irreversibly, which means they permanently bind to the active site of an enzyme. And if that's going to happen, then the enzyme is out for good. The enzyme can't catalyze the reaction. And if you've got a lot of competitive inhibitor in your reaction tube, your enzymes won't be able to catalyze the reaction rate should slow down. Non-competitive inhibition also slows the reaction down, but a non-competitive inhibitor, and here we've got kind of an orange blob, binds to the enzyme, that's our big purple blob, at a place away from the active site. But what it does when it binds is it changes the shape of the active site. And here you can see the active site is sort of changed shape. It looks now like its thumb is sticking instead of up, it's sticking out, right? When that active site changes shape, then the substrate can't bind. So a non-competitive inhibitor slows the reaction rate down because it binds to a site somewhere on the enzyme and changes the shape of the active site. Here's a good example of a competitive inhibitor. Maybe you've heard of DFP or sarin. These are both nerve gases, and they are actually both examples of competitive inhibitors of an enzyme called acetylcholinesterase. So I just like this example because it's just fascinating. DFP is a nerve gas. If it's inhaled or contacted through the skin, it can cause all sorts of problems. So you might first have sweating, vision problems, vomiting, convulsions. Eventually, you will have bronchial constriction or paralysis of the respiratory system, and you die by asphyxiation. You can't breathe. And it turns out that DFP is a competitive inhibitor of an enzyme. That enzyme is called acetylcholinesterase. Now, in order to figure out what this enzyme does, let's take a look at this picture here. Here we've got two neurons. We could also think of this as being a muscle cell. Let's do this one as a muscle cell. A neuron interacting with a muscle. All right, a muscle cell. And what this neuron does to tell the muscle, hey, it's time to contract now, is it releases a neurotransmitter called acetylcholine into a gap. That gap is called the synapse, and the neurotransmitter acetylcholine binds to the muscle cell, and it tells the muscle cell, hey, contract now. But in our muscle system, in order to do anything, our muscle cells need to be able to contract and then relax, and contract and relax all the time. And so our nervous system has to have a way to get rid of the extra acetylcholine, or otherwise our muscle cells would just stay contracted. That would be bad. We'd be paralyzed. Normally, there's an enzyme called acetylcholinesterase that recognizes acetylcholine and breaks it down. And the parts of acetylcholine are recycled. They're returned to this neuron and then recycled. DFP is a nerve gas that binds to acetylcholinesterase and prevents it from functioning. If this doesn't work, we get a buildup of acetylcholine inside the synapse, causing our muscle cell to stay permanently contracted. And this happens in our lungs, and our lungs can't move, they can't change shape, and we can't breathe. And that's horrible. So that's just an example of a competitive inhibitor of acetylcholinesterase. Okay, there's another type of regulation that's worth mentioning. It's called allosteric regulation. I'm not going to do that part. Allosteric regulation involves non-competitive molecules, okay? And what we've got here are an enzyme called an allosteric enzyme. Allosteric enzymes are kind of complicated. In our picture, we've got this purple enzyme that has kind of 4 different parts put together. And allosteric enzymes can change shape. The allosteric enzyme in this picture is changing shape by, it can either be in its active form or its inactive form. And so allosteric enzymes, the word actually means other form, other shape. It can change shapes. Okay? This particular allosteric enzyme has 4 active sites to which the substrates can bind. And in its active form, those active sites are available for substrates to bind to it. In its inactive form, the active site changes shape and no longer, this enzyme can't work. Now, there are some molecules that are considered activators, allosteric activators, bind to the enzyme and stabilize it in its active form. What's going to happen to the rate of the reaction if there are activators present? Well, the reaction rate should increase because the enzyme is always in its active form, catalyzing reactions. On the other hand, there are also allosteric inhibitors that bind to the enzyme and stabilize it in its inactive form so it can't catalyze reactions. So if you've got an allosteric inhibitor in your reaction tube, it's going to bind to your enzyme, keeping it in its inactive form, and the reaction rate should definitely drop. It should inhibit the reaction. So allosteric enzymes are special enzymes that can change shape. They can oscillate between active and inactive forms, and there are allosteric activators, allosteric inhibitors that can change the rate of the reaction. One last topic in this chapter, and that is a return to that term of metabolic pathways. Now, it turns out that metabolic pathways, remember we had a pathway at the beginning that went something like this. We want to convert A into D. Each reaction requires an enzyme, enzyme 1, enzyme 2, enzyme 3. Metabolic pathways are regulated by enzymes, by their concentration and by their activity level, whether they're active or not, right? And many of our metabolic pathways have what's called feedback inhibition. Feedback inhibition. Let's check out what feedback inhibition means. Here's my drawing of that fake metabolic pathway that we were just talking about. Now, let's say that a cell wants to make D, and wants to make a certain amount of D. You don't want to make a ton of D, but you need to make enough. Okay? Now, in an example of feedback inhibition, D, the product D, the molecule D, can feed back on enzyme 1 in this feedback, in this pathway, and feedback in a negative way. Maybe D is an allosteric inhibitor of enzyme 1. Okay? So if there's a lot of D around, what's going to happen to enzyme 1? Enzyme 1 is going to be inhibited. Okay, so in this particular reaction, let's say there's a lot of D. There's just an abundance of D around. What's going to happen to this metabolic pathway if there's a lot of D? Well, D is going to feed back on enzyme 1 and inhibit it. And if enzyme 1 doesn't work, what happens to the whole pathway? It shuts down. But then there's not very much D. Then the level of D goes down, and what happens to the pathway? Well, with not much D, there's not going to be much to feed back on enzyme 1. Enzyme 1 should function, the reaction pathway should work, and we should get more D. This is a nice way for a cell to make enough D, whatever D is, and if there's too much D, to shut the pathway down. Our cells have all sorts of metabolic pathways that are regulated by feedback inhibition. This is a fantastic way for a cell to make enough of something, but not too much. You don't want to make too much of something that you don't need to make too much of. That's a nice setup. there are actual examples of feedback inhibition. This is one of them. You can check out figure 8.21 to take a look at how feedback inhibition is used to regulate the production of an amino acid called isoleucine. Okay? And we see that isoleucine, the end product, is an inhibitor of an enzyme early in the reaction or in the pathway. So that typically is what happens. A product late in the pathway inhibits an enzyme early in the pathway to shut that enzyme pathway down if there's a lot of the end product available. And then there are a series of review questions. You can work through these on your own. Some of these will show up on the assessment for this module as well. And that's the end of this chapter. And so we'll finish up here.